Bismillahirrahmanirrahim. Assalamu alaikum and welcome to Sindh Biotechnology Association. In today's lecture, we are going to discuss about the DNA recombinant technology. As very clearly name suggests, recombining, recombination of DNA. In recombinant DNA, the molecules of DNA from two different species that are inserted into host organisms to produce new genetic conditions that add value to science, medicine, agriculture, and industry. The definition of recombinant DNA technology that it is a series of procedures that use to recombine DNA segments. Under certain conditions, a recombinant DNA molecule can enter a cell to replicate. That means it we allow it to clone itself. Since the focus of all genetics is the gene, the fundamental goal of laboratory geneticists is to isolate, characterize, and manipulate genes. Although it is relatively easy to isolate a sample of DNA from collection of cells, finding a specific gene within a DNA sample can be compared to finding a needle in haystack. Considering the fact that each human cell contains 2 meters 6 feet of DNA. Therefore, a small tissue sample will contain many kilometers of DNA. However, the recombinant DNA technology has made it possible to isolate one gene or another segment of DNA, enabling the researcher to determine its nucleotide sequence, study its transcript, mutate it in highly specific ways, and reinsert the modified sequence into living organism. The history of recombinant DNA technology, it is very recent and advanced in biotechnology. It is developed by two scientists named as Boyer and Cohen in 1973, and later in 1980 they both are awarded for this discovery. The basic principle The basic principle of recombinant DNA technology is that the DNA is inserted into another DNA molecule, that is its vector. The recombinant vector, that is having both DNA, a foreign and of itself. It is then introduced into the host cell where it can clone itself. It can make copies, replicate itself, and then the gene is produced. In this diagram, we can see that how we have taken a specific part of a gene, we cut it through endonucleases. Here is a specific enzyme that recognizes a specific base sequence in both plasmid. Plasmid is an extra chromosomal unit of this bacterium and chromosome and cut it the DNA. After that, another enzyme joins the open plasmid and then the specific enzyme is inserted into plasmid DNA section and then we allow it to clone itself. Its application, it has very large diversity in field of its applications. It helps in the growth, in producing growth hormones, insulin, interferons, blood clotting factors. We can produce a large scale human proteins by genetically engineered bacteria. In today's lecture, we will see that how can we produce human insulin through this technology. The number one step is to obtain the human insulin gene. The human insulin gene can be obtained by making complementary DNA, cDNA, copy of messenger RNA for human insulin. On the other hand, we just took the bacterial cell and cut its plasma out with restriction enzymes. One side we have cDNA from human pancreatic cell, that is complementary DNA made from RNA using reverse transcriptase. On the other hand, we are having a plasmid which we have cut it through restriction enzymes. In second step, what we do, we join human insulin gene into that plasmid vector which I have already cut it. The bacterial plasmids and the cDNA are mixed together. The human insulin gene, cDNA, is inserted into that plasmid through complementary base pairing at sticky ends. Step 3. Introducing the recombinant DNA plasmid into bacteria. Now, that recombinant DNA that having cDNA of insulin and that plasmid, now we will introduce into a bacteria. E. coli is used as a host cell. If E. coli and the recombinant plasmid are mixed together in a test tube. Step 4. Selecting the bacteria which have taken up the correct piece of DNA. The bacteria are spread onto the nutrient agar to provide important conditions and nutrients so that it may grow. The agar also contains substances such as antibiotics which allow the growth of only transformed bacteria. How it is helpful in gene therapy for genetic diseases. It is still in experimental stages. It may be possible to transfer the gene for normal adult hemoglobin into narrow stem cell of an individual with sickle cell anemia. The goal is to promote the growth of enough cells to produce enough normal hemoglobin to alleviate the symptoms of sickle cell anemia. 100% is not required to attain the elevation of symptoms. There are few safety issues related to recombinant DNA technology. As bacteria is commonly used in recombinant DNA work, there has always been a concern among the scientists and a worry among the people that there is a possibility that a clone of highly pathogenic recombinant bacteria were made by accident, then escaped from the laboratory and caused an epidemic for which no drugs were available. Recombinant DNA Advisory Committee RAC was established in 1974 in US, which responds which responds to public concerns regarding the safety of manipulation of genetic material through the use of recombinant technology. There are two types of contaminant control, biological and physical. Physical contaminant. It is very effective biological safety program that were operated in a variety of laboratories which include a set of standard practices generally used in microbiological laboratories and special procedures, equipment and laboratory installations that provide physical barriers of varying degrees. In considering biological contaminant, the vector, that is a plasmid, it can be organelle or virus, for the recombinant DNA and the host that can be a bacteria, plant or animal cell in which the vector is propagated in laboratory will be considered together. A survival of the vector in its host outside the laboratory and the transmission of the vector from propagation host to other non-laboratory host. Dangers of DNA recombinant technology. It is always possible that an antibiotic resistant plasmid could accidentally incorporate it into a dangerous pathogen with the serious medical consequences. So that's all for today's lecture. Thank you so much for your patience.